Hello everybody, this is Mirko Rossetti, product specialist of Milestone, and I'm pleased to welcome you to this webinar, which is focused on the single reaction chamber digestion applied to food samples. So if you have some experience with the sample preparation of food for elemental analysis, you know that today trace metal analysis for food samples is something pivotal, is a pivotal task, and is usually run to comply with legal requirements. There is also a growing awareness and concerns about the food safety, which is resulting in tightening of the regulation for those that we know as toxic elements, including arsenium, cadmium, lead, and mercury. So this webinar is going to be split in three main sections. So the first section that we will see is related to the main challenges that you can face during sample preparation of food samples and how we can address them. So let me first of all clarify that if you are familiar with digestion of food samples, you know that probably the first one of the biggest challenge is to meet the very low limit of quantifications related to the analysis of food samples, but we have also to consider the difficulties in getting a complete digestion. So sometimes the digestion of food may lead to incomplete digestion, affecting then the results of analysis, as well as the challenges in facing the reactivity of food samples. As we will see later, some of them can be quite reactive, releasing a lot of pressure during the digestion. So this pressure must be properly handled by the digestion system, as well as the difficulty to prevent any contamination from many potential sources that can affect the final analytical data. So we can say that generally, a successful analysis strictly depends on the quality of digestion. So high quality of digestion lead to a high quality of analysis. And vice versa, bad quality of digestion, so incomplete digestion, can lead to low recoveries of the target elements, as well as to some interferences that can affect the analysis. Having said that, so we can say that method development is extremely important when digesting your food samples, and during the optimization of the method to find a proper method to get complete digestion, you have to consider three main players, three main parameters, which are the reactivity, strictly connected to the sample mass, the chemistry, meaning the acid mixtures involved in the digestion process, and the digestion working conditions in terms of mainly temperature and pressure. So let's go in details and see one by one all these parameters. So the first one is related to the reactivity. First of all, let me clarify that when we talk about food, we have to say that food incorporates a very large variety of matrices that can be classified usually according to their activity in three big classes. So the first class of food compounds incorporate what we call the fresh sample. So sample having a very high content of water, like fresh meat, vegetables, fruits and fruit juices, fresh fish, and so on, which are usually sample non-reactive, or anyway with a very, very limited reactivity. As a second class, we find what we call the dry sample. We can consider like dry, or anyway sample with a very low content of water, which usually have a medium reactivity, like rice, pasta, cocoa, chocolate, baby food, milk powder, and these kind of products. And then as the last family of food compounds, we have what we commonly call the high energy containing samples, which are oily, fatty samples, so all these kind of samples containing, having high fat, oil, sugar, and alcohol content. So here we can find oils, alcoholic beverages, we can find honey, sugar, which are generally very high reactive samples. So this is the common classification of food samples, but let me also say that the heterogeneous nature of most of the food compounds make the classification into a unique class of compounds very tricky in most of cases. So, and that's why the digestion system selected for a sample preparation of food must be properly able to hold up the temperature and pressure conditions developed during digestion, especially for those samples that are more reactive than initially expected. So, as I said, the reactivity is strongly connected to the sample mass. The sample mass is one of the most important parameters to define, and usually it is set according to the instrumental detection limit, and in order to make sure that the portion of sample selected is as much homogeneous as possible, just to make sure the analysis is reliable, and so we have to set an homogeneous portion of sample. Typically, for food samples, the typical sample mass is selected up to half gram, which is a good compromise to match the detection limit and to ensure a good homogeneity of the sample. Say that, of course, it's possible to increase the amount of sample, just always considering that increasing the amount of sample allow us to lowering the detection limits, to achieve lower detection limits, but also considering that the higher the sample mass, the higher the pressure developed during digestion. So always consider that the digestion system that you are going to use must be properly able to hold up the pressure developed during the digestion. So the pressure usually depends not only by the sample mass, but it also depends by the reactivity, the sample type, and its reactivity. That's why the method development that you have to adopt for highly reactive samples has to consider some specific approaches. So usually when dealing with highly reactive samples, I'm used to recommend working with a very limited amount of samples, at least for the first stage. What I mean is that when you have to digest reactive samples, or anyway unknown samples where you don't exactly know the reactivity, but you expect a quite high reactivity, my suggestion is to start testing 100 or 200 milligram, and then step by step increase the digestion in case you see that the pressure developed is not really high and anyway can be properly handled by the system. Then another good practice when dealing with highly reactive samples is to make the heating a bit longer. So just extending a little bit the ramping time, just 5 or 10 minutes is more than enough usually. And this is just to allow the sample to be heated up, warming up gently in order to prevent the reactive samples from reacting violently. And then another important recommendation that we are used to share with our food users are the capability, the possibility to add a small amount of water, usually 1 or 2 ml are more than enough, just to limit to adding the ionized water to the current acid mixtures allow to limit the strong reactivity of the sample. Okay, when then the second parameter to consider for the method optimization and method development of your food samples digestion is related to the chemistry. So the acid mixtures involved in the digestion. Generally, the digestion of food samples always involve the use of nitric acid as a main player, which is then often used in combination with some other reagents. For instance, hydrogen peroxide is usually recommended because it's a strong oxidizing agent that can help the complete dissolution of the sample. And this is usually recommended in a small amount, like usually in ratio 4:1 with nitric acid. So 1 or 2 ml of peroxide are a good recommendation. Or sometimes also hydrochloric acid is recommended in a very small quantities, I mean few drops, just to stabilize some elements, especially for the volatiles. Say that talking about the acids and acid mixtures, let me also add that as you know, there are different quality and different purity acids available on the market. Of course, the grade of quality and grade of purity should be selected according to the detection limit that you want to achieve. The lower the detection limit you need to satisfy, the higher the grade of purity and quality of acid that you should use. That's why for a trace metal analysis, high quality acids, at least ultra pure or ultra trace analysis acid must be selected. This because it's very important to minimize as much as possible the interferences, to improve the sensitivity, and at the end to get accurate and reproducible data. The third parameter to play with during the method development is related to the digestion working condition, especially in terms of temperature. So the working temperature strictly depends on the type of sample, the specific type of sample that you are going to digest. Generally speaking, for food samples, the typical working range of temperature is from 180 up to 250 degrees C, depending on the sample, considering that the higher the temperature, the higher the quality of digestion, because the higher the temperature, the lower the residual carbon content that you have at the end of digestion in the solution. Okay, so once we have seen what are the main challenges in the sample preparation of food, we move now onto the next section, which is related to how the single reaction chamber technology applied to the digestion of food samples can improve and elevate the sample preparation process. So firstly, let me do a very short introduction. So as you probably know, there are many different kind of techniques that can be adopted for sample preparation of food. Honestly with you, the most efficient and most commonly used is the microwave-based technique. And because of all the benefits involving the use of microwaves. And let's say that Milestone has a huge experience in the microwave sample preparation field. That's why Milestone has developed over the years a large number of microwave-based technologies that can be classified in two main categories. So on one side, we have the rotor-based technology, and on the other hand, so we have the single reaction chamber technology. Both of them are working with microwaves, but for the purpose of this webinar, let's now focus only on the single reaction chamber technology. So let's see in details why the application of the single reaction chamber technology allows to improve and elevate the sample preparation of food samples. So the first benefit in using the single reaction chamber technology for food is related to the unmatchable performance in terms of temperature and pressure, related to the fact that SRC technology allows an excellent dissipation of heating in case of strong exothermal reaction due to high sample reactivity. Then Of course, we have also to consider the capability of the system to handle very high pressure up to 199 bar. That can be developed during the digestion of reactive samples, as well as the capability of the system to work at extremely high temperature, up to 300 degrees C, so much higher comparing to the traditional system, which allow to improve the quality of digestion and reduce the interferences during the analytical step, because, as I said before, higher temperatures allow to get a higher quality of digestion because of the lower carbon content in the final solution, which will not affect at the end the analytical step during ICP analysis. Then, applying the single reaction chamber technology to the sample preparation of food allow to work with larger amount of sample. So as I said before, the typical sample mass selected for digestion of food is around 0.5 gram. The single reaction chamber technology allow to increase this amount up to more than 2 grams in each digestion cube, working with very small amount and volume of acid, just a few milliliters. This is a great benefit because the limited volume of acid allow to reduce the consumption of acid as well as the waste generation, and allow to limit, to reduce the analytical blanks and the dilution factor in order to better match the detection limit you need. As well as very important, we recently have developed with the single reaction chamber technology some methods working with diluted acid. So usually it's diluted acid 1:1 in the ratio 1:1 or 1:2 with the ionized water, which allow simply by increasing a little bit the final temperature. So at the end, we can further save acids. Then the single reaction chamber technology is the unique technology available on the market, which allow to run mixed samples batch. So it means that because it's the unique technology that allows to get exactly the same conditions in all the digestion vials. So this means basically that you can collect all the samples that you have, and you can mix completely different samples in the same run, regardless the type of sample, regardless their activity, the sample amount, the volume of the acid used, and so on, regardless any kind of working conditions. So at the end, it means that you will not need anymore to run separate cycles just for the reactive samples separately than comparing to the rest of the sample. You can run now all of them together. Last but not least is the extreme capability of the SRC technology to improve, to elevate the throughput and to increase the overall productivity of your laboratory. So if you look at the table in this slide, you see that this is something that we have did with our UltraWave 3 single reaction chamber technology, equipped with a 20 position rack. So basically what we have done during this study is just to estimate the total operator time required for each single step involved in the sample preparation, starting from the beginning, so from the weighing of the sample until the final solution, until getting the final solution ready for ICP analysis. So basically, as you can see from this table, we set a microwave program, which is a common microwave program used and suggested for food samples at 200 degrees C. The total running time of the microwave program is 30 minutes, 20 of ramping and 10 of holding time. Of course, this is a very general microwave program. As I said a few slides ago, the the digestion conditions should be adjusted properly according to the specific type of sample. It means that this program can be further shortened, and the final temperature can be slightly increased to 120 up to 240 degrees C, depending on the specific sample that you want to digest. This is a very general one. Anyway, what I want to show you is that the single reaction chamber technology allows to extremely simplify the handling. If you look at the column related to the vials assembling and disassembling, you see that no time is required for the operator for the assembling and disassembling. And this is because the SRC technology allows to work with loose fitting caps, so there's no need, comparing to the traditional system, to hermetically close each position one by one. Plus, consider that the venting is not considered in this step. I mean, normally with the commodity systems, with the traditional digestion systems, you need to wait a certain time to let the vapors to be released by the tubes. So this is what we commonly call the venting under the fume hood, and this should be done very slowly for the safely release of the vapors. This is something that it's not absolutely required with a single reaction chamber systems, because the release of the vapors is included in the pressure release, which is done automatically by the system during the cooling. Then, considering the digestion cycles, they are extremely fast. As I said, heating is very fast. It's usually around 30 minutes or even less. And the cooling is even very fast, because it's done by water. So the step including cooling plus release of pressure takes approximately 10 to 12 minutes, not more than this. And plus, if you look at the table, you will also see that there is nothing related to the cleaning. This because the single reaction chamber systems allow to work with disposable glass vials, which do not require any kind of cleaning at all. So using glass vials, we can completely skip the cleaning, so we can use once and then waste directly. At the end, considering all these steps, we can say that we can, starting from the beginning until the end, so from the weighting of the sample until getting the final solution ready for analysis, we can prepare 20 samples, so we can digest up to 20 samples in approximately 55 minutes, so in less than 1 hour. If we extend this productivity for the 8 hours of a typical working day, we find that we can do more than 8 runs per day, which means up to more than 170 samples digested in a single working day, which is really a huge number. So the unmatchable productivity of the single reaction chamber technology. Okay, we move now into the last section of this webinar. So after seeing why the application of the single reaction chamber technology to the food samples allow to simplify and elevate the sample preparation process, we go now in details through this practical example related to the sample preparation of baby food samples for trace metal analysis. Firstly, let me say that if you are familiar with baby food digestion, you know that baby food is a very complex matrix because it includes a large variety of components, including proteins, vitamins, carbohydrates, fats, and much more components, making basically this type of food usually very reactive. That's why the proper digestion of baby food can be quite challenging. So not only because we need usually to match a very low limit of quantification, so we are talking about sub ppb or ppt levels, but even because of the unknown reactivity that potentially may lead to exothermal reactions, and because of the unknown composition of these samples that can lead to low quality of digestion, so usually sometimes incomplete digestion that can affect the final results. Also, always considering that during baby food digestion can lead to, baby food challenges during sample preparation can be also related to the matrix effects that can cause over or underestimation of the signal during analysis, as well as may be affected by external contaminations. Having said that, the trace metal determination of baby food is nowadays extremely important. There is a particular concern around the baby food toxicity, and this is more evident for baby food because babies and children are the most vulnerable to the neurotoxic effects of what we call the toxic elements that we have seen at the beginning of this presentation. These lead to very strong and tightened regulations about the toxic components and to intensification of the monitoring, of the checking of all the ingredients involved in the baby food components. So that's why back in 2021, so very recently, the US Food and Drug Administration has announced the Close to Zero initiative in order to strongly reduce the exposure of the toxic elements in baby food as much as possible to reach ideally the zero value, so to go very close to zero. So basically, this slide show is a clear picture of what's the US FDA current situation. So at the moment, relating to the reference method, which is the FDA AEM 4.7 method for food and related products. So if you look at the table, you see that we have the 4 toxic elements listed in the table. So arsenium, cadmium, lead, and mercury. And for each of these elements, we can see the limit of quantification of the FDA in microgram per kilogram and the related BFSA. So the last column, the BFSA in microgram per kilogram, represents basically is the Baby Food Safety Act, which represents the current limit according to the regulations for the toxic elements in baby food. So it's clearly evident that if you compare the second column, so the FDA LOQ at the moment with the BFSA, the last column, you see that for most of these elements, the limit of quantification of the FDA are actually above the limit set by the BFSA. And that's why FDA is putting all its efforts in order to strongly reduce its limit of quantification for the toxic elements to match the BFSA regulations. So referring to the methods, we have seen that the reference method is the AEM 4.7 for food and related products, and this method actually provided 2 separate approaches for sample preparation. So we have on one side we have the closed vessel microwave approach, which is basically the rotor-based traditional approach, and on the other side we have the autoclave style microwave approach, which is equal to the single reaction chamber. By the way, just consider that US FDA is one of the largest, or probably I have to say the largest adopters of single reaction chamber technology over the years, including many ultrawave and ultraclave units from Milestone for their food testing labs. This is something that we are so proud of, and we want to share with you this very important and useful information. So basically the autoclave style microwave approach is something that they have developed using our SRC technology, and as you can see, thanks to the higher temperature that they can achieve with the SRC. So 250 compared to the 200 degrees C of the traditional approach. And thanks to the limited lower volume of acid, so it's just 5 ml nitrate plus 1 ml peroxide with the SRC, with the autoclave approach, comparing to 8 ml nitric and 1 ml peroxide with the closed vessel approach. We can say that using this autoclave style microwave approach, they have been able to lower the limit of detection and the limit of quantification as well. What we did in this study basically is simply adjusting a little bit the reference method used by FDA and applying it to our new UltraWave 3. So thanks to the highest capability of the UltraWave 3 and highest performance, we have been able to further reduce the limit of quantifications that comparing to the LOQ that FDA is getting at the moment, simply by increasing the sample mass and reducing the volume of the acid. So the table that you see in this slide is exactly the same that we have seen before, except for the last column, which is related to the limit of quantification that we have been able to get with our UltraWave 3 system. So simply by modifying the reference AEM 4.7 method, so simply increasing the amount of sample and reducing the volume of acid, as you can see from this table, we basically have been able to strongly reduce the LOQ really close to the zero. So, as for your information, the method that we have been adopted for to get these great results is based on digesting baby food with the UltraWave 3 equipped with the 20 position rack, so working with 20 samples simultaneously, 1 gram of sample per each tube, digested with only 5 ml of nitrate up to 250 degrees C and diluting up to 50 ml. So with a dilution factor which is reduced to 10. So if you want to learn more about the study that we have done on baby food in this scenario with FDA, I strongly invite you to have a look to our ebook for baby food that you can freely find and read from our website. In this ebook, I want to point out that you will find the milestone recommended approach that allow us to significantly lower the limit of quantification, and you also will find the overall approach that we have adopted for the sample preparation from the beginning until the end, because you have to know that the data that we have been able to get related to the close to zero limit of quantification have not been obtained simply focusing on the digestion step. So this meta-analysis is not only because of the performances of the UltraWave. This is an extremely important step, of course, but it's something related to the overall sample preparation workflow. So in this ebook, you will find how we have been able to properly clean all the vials at PPT levels thanks to the milestone trace clean, so the acid steam cleaning apparatus. You will find the references to the easy fill, so the milestone automatic dosing stations to make the addition of the reagents totally, completely automated. And then, for instance, you will see that the acids that have been used for these purposes, for this study, have been prepared by using the duo pour system of milestones, the sub-boiling distillation system for the homemade production of ultra pure and ultra trace acids. So this is to say that Milestone takes care not only to the digestion step, but to the whole sample preparation step from the beginning until the end. And so we are now proud to say that we can provide a full range, a full portfolio of products, enabling to simplify and make the sample preparation as much automated as possible in order to improve the analytical blanks and the productivity of your lab. Okay, so just to conclude, I want to refresh you the main reasons why the single reaction chamber applied to the sample prep of food can elevate the sample prep, the overall sample prep step to a further, a higher step. So we have seen we have talked about the highest, the unmatchable performance of ultrawave of this SRC technology in terms of temperature and pressure comparing to the traditional system. We have seen how the SRC allowed to strongly increase the sample mass to match lower detection limit, and how we have been able to further reduce the acid volume, which means save acid and reduce the waste, basically. Then we have seen the unique capability of the SRC systems to run mixed batch samples, so to batch completely different kind of samples and run all of them together with a unique method without needing of batch separately specific type of samples. And then we have seen as the last point, but very important, how the SRC allow to improve the overall throughput and productivity of your lab. Okay, so I think that's all. I hope everything is clear. So thank you for participating to this webinar, and goodbye.